So the properties or characteristics of lac. Lac is soluble in alcohol and weak alkalis. It has the capacity to form uniform durable films. It is highly uh, uh, stretchable. It has uh, scratch hardiness. That means it doesn't get scratches. It has resistant to water. It is a good adhesive and good sealer, and it can allow quick rubbing without forming slicking or gumming. So due to this many different characteristics, it is used as a multi-purpose resin. Chemically, it is a mixture of 50-50% mixture of hydroxyaliphatic acids and sesquiterpenoic acids, and the lac consists of 50% uh, consists of around 68% of resin, 6% of wax, 1% of dye, and rest of the things are around 25%. Now let us see the differences between uh, adult, the male and female. So the female uh, is oval, having a pair of small branchial pores in interior side and a single round and anal tubular opening in the posterior side. Through the anal tubular opening protrudes waxy white filaments secreted by the glands in the insect body. So these white filaments are indication that the insect is live inside the uh, in this resinous cell. So and it is in healthy condition. So these filaments also prevent the blocking of the pore during the excess secretion of the lac. An adult male is usually winged. It is larger in size and than female, and it is red in color. The body is typically divided into head, thorax, and abdomen. The head bears a pair of antennae and a pair of eyes. Mouth parts are absent, so the male adult insect is unable to feed. Now, life cycle of lac insect. As I told earlier, Lucifer lecca is the main species, but Caria lecca is also there, which is used for lac production. The life cycle of insect takes about 6 months and consists of stages: egg, nymphal instar, pupa, and adults. The adult male is winged and walks over the females to fertilize them. The female increases in size after fertilization to accommodate her for laying number of eggs. After 14 weeks, the female shrinks in size, allowing light to pass into the cell. Cell means the coating of this lac. Its own body ultimately gets covered with lac, so this is called cell. And this so when the body of the lac insect, female insect shrinks, uh, it allows the light to go inside, as well as it creates a space for laying of the eggs, and this happens after around 14 weeks. So lac is secreted by insect for protecting itself from the predators. The female oviposits a large number of eggs in the space called ovisac, which is between the surface of lac insect and the cell. So when the eggs hatch, the larvae emerge and the whole process begins all over again. Now, how what are the steps in the commercial production of lac? During leg cultivation, proper care of the host plant, the propagation, uh, proper care of the host plant, propagation process, and collection of the leg are the important steps. So, uh, if this can be divided in the 5 steps. One is selection of the site and the host plant, pruning of the host plant, inoculation. Means releasing of larvae, small larvae, uh, onto the this host plant, and that is done by tying a small stick which is called brood leg, so where the eggs have just hatched and the larvae have emerged. And swarming means uh, this so after so this inoculation can be done just before hatching of the eggs. So after hatching, the larvae emerges uh, from this eggs, and they will go to other uh, shoots of the plant to uh, find their food, and so they will stick to the tender shoots of the host plant. So this going and infesting other new branches of the host is called swarming, and then harvesting after finally lac is secreted by the adult insects. And important host plant of the lac are kusum, palas, bear, babul, kher, and arhar. Kusum is called Sclera trijuga. Palas is Butea frondosa. Bear is Jujubas, Jujuba. Babul is Acacia arabica. Kher is Acacia cachicu. And Arhar is Cajanus cajan. Lac insect thrive on more than 400 species that grow in the forest, and these trees can have many uses. So lac pest. So the the danger of lac cultivation is the infestation of the attack on the lac insect by predators and parasites. So around 22 species of lac predators are there, 30 species of primary and 45 species of secondary parasites, and several fungal and other pathogens are there. Cultivation. The cultivation of lac involves proper care of the host plant, regular pruning of the host plant, infection or inoculation, crop reaping, control of the insect pest, and forecast of the swarming, collection, and processing of lac. The first important prerequisite for the cultivation of lac is to take care of the host plant. Uh, so when the host plant reaches a particular height, then it should be used. It's the larvae feed on the cell sap by inserting their proboscis into the tender twigs of the host plant. The proboscis can only be inserted in tender uh, offshoots, so the pruning is required so that new branches may emerge. For this, before inoculation, uh, the pruning is necessary. So the branches less than 1 inch in the diameter are selected for pruning. So the branches which are around half inch in diameter, half inch or less, they can be cut from the base. If the branches are bigger, wider in the diameter, more than half inch, then they should be cut around 1.5 inch above the base of the branch. Now, inoculation, the method by which the lac insect are introduced to the new lac host plant is known as inoculation. The infection from one plant to another occurs by natural means also, but then it is not very beneficial as far as lac cultivation is concerned, and that is called natural infection. Natural infection takes place due to the overcrowding or non-availability of tender shoots on the particular tree. So artificial infection is done by the cutting a branch on which eggs are there, and that is called brood leg. It is around 6 inches in the length. So brood legs are kept for about 2 weeks in some cool place, and when the larvae start emerging from those brood leg, it is ready for inoculation. So it should then be tied to the host plants. It can be tied longitudinally, it can be tied laterally, or it can be tied intercalated. That means it can be tied among many uh, shoots or many tender shoots of the host plants. Leg crop. So leg insect repeat its life cycle twice in a year. There are actually four leg crops since the leg insect behave in two ways. Either they develop on kusum plants or develop on the plants other than kusum. The leg which grows on non-kusum plants is called Ranjim leg or Rangini, and which grows on kusum plant is called as Kusumi leg. Four leg crops have been named after four Hindi months in which they are cut from the tree. They are Katiki, Rangini crops and Kusumi crops. So Rangini crop is further divided into two. Katiki, that where the lake larvae are inoculated in the month of June, July and reaped in Kartik, that is October, November. So when they are reaped uh, in Kartik month, they are called Katiki, and when they are reaped in Besaki month, Besakh, that is April, May, then they are called Besaki. So larvae produced by Katki crop are inoculated in by larvae produced by Besaki crop are inoculated in October, November and reaped in Besak, that is April, May. Kusumi have two types, Aghani and Jethoi. Aghani is earlier, so Aghan months. So this is reaped in December, January, and lake larvae are inoculated in June, July. Jethoi is reaped in Jest month, that is June, July. So the larvae produced by Aghani crop is inoculated in the month of uh, larvae produced by Jethoi crop is inoculated in the month of January, February. Male emerges in the month of March, April. Female give rise to swarming larvae in June, July, and the crop is reaped in the month of Jest, that is June, July. So it is Jethoi. Now type of lac, it is button lac, seed lac, stick lac, and shell lac. Stick lac is when it is obtained from the when it is scraped from the sticks, or when it is there on the stick also it is called stick lac. Then it is processed to form seed lac. Then it is further processed to make button lac and finally shell lac. Scraping and processing of lac. Lac cut from the host plant is called stick lac, airy lac, and funky lac. Airy lac is collected before the emergence of larvae, and funky after swarming has taken place. After scraping, lac should not be exposed to sun. The scraped lac is grinded in hard stone mills. The unnecessary material are sorted out in order to remove the finer particles of dirt and color. This lac is washed repeatedly with cold water. At this stage, it is called seed lac. Seed lac, after melting, is sieved through the cloth and is given the final shape by molding. The final form is called shellac. So when it is sieved from the plant, from through the cloth, then it is put in the form of small button like shape. So then it is called button leg, and button leg is further process to get shellac. There are different colors of the leg can be obtained by mixing with the different chemicals. Leg enemies and uh, leg enemies are parasites and predators. The common parasite of leg insect is known as chalcid. They are small winged insects. They lay their egg inside the leg coat, either on the body of the leg insect or inside the body of the leg insect. The larva which hatches from these eggs feed upon the leg insect, thereby causing mortality of their host. Damage done by this parasite contributes about 5 to 10% of the total destruction of the leg crops. Predators cause more destruction around 35%. Eublema amabilis is called white moth, and Holococera pulveria is called blackish grey moth. These are important predators. They do not feed only not only feed on the leg insect but also destroy the leg. Other parasites are Tecardiae, Fagus tecardiae, and uh, Aprostocetus uh, purpureus, or Aprostocetus is also called Tetrastricus. So these are the two important parasites, and they lay their eggs in the leg cells, and the grub, that is their larvae, uh, hatch out and feed on the leg insect within its cell. And next is predators. Predators are Eublema amabilis and Pseudo Hypetopa pulveria. These are two important predators, but there is also one which is called blackish grey moth and Holococera pulveria. Now, control method, there are cultural control, artificial method, chemical method, and biological control. In cultural method, the brood leg showing the minimum enemy should be selected for inoculation and should be cut from the host plant very near to the time of emergence of larvae, and that is around 1 week before the emergence. The brood leg used for the inoculation should be removed from the new host plant branches as soon as the emergence of larvae, that is approximately 3 weeks after inoculation. So after suppose this hatching has taken place, after that hatching 2 weeks we should wait so that larvae go to the other branches, and then we remove the old branch which was used for inoculation. The leg should be scraped at once and rest should be crushed or dropped into fire in order to destroy the predators. So this old broad leg branch, after it has been used for inoculation, then after removing it, it should be uh, destroyed. The delay in processing so after harvesting, if there is a delay in the processing, that also gives chance to the uh, enemy insect to escape into the fields. So the manufacturer should try to convert stick leg into seed leg as soon as possible. Artificial method is to convert the huge amount of stick leg to the seed leg. It takes time, so to avoid that damage during this time, the stick legs should be tied in the bundles and should be tied with stones and immersed in the water in the river or pond for about a week. Chemical method is the use of pesticide and biological method hyperparasites. The chemicals in the chemical management IPM can be used and ethofenprox, carbosulfan, indoxacarb and fipronil etc. These can be used in IPM. Biological control trichogramma, trichoexigum and trichogramma RK, trichogramma exigum, trichogramma austrainae. They are able to they are hyperparasites of Eubulima amabilis. Then the reduction in one study, they found that with the use of this uh, hyperparasites of around 20 eggs per bush, around 70 to 86% reduction in the Eubulema amabilis was observed in the Rangini crop and about 52 to 70% in the Kusumi crop. Uh, so this is all